In this video, I'm going to show you how to reconstitute your lyophilized stem cell exosomes. We are going to start by taking the cap off of the bottle. It just pops right off like so. And then I'm going to take an alcohol swab that I provided for you, and we're going to just clean off the resealable cap on the bottle itself. Then we're going to take the bottle of the syringe of saline, We're going to attach the hypodermic needle that I've included, so unscrew the cap here, and then very carefully we're going to attach the needle to the syringe like so. Once that's secure, we want to carefully remove the lid off of the needle. Next up, we are going to push out the extra air out of that syringe, and then we're going to push the needle through the center point of the cap on your exosomes. Now, those exosomes are going to pressurize, so what we want to do is pull back on the syringe to take some of the pressure out of that sealed bottle. Then we're going to pull it out and push the excess air out of the bottle. This is going to be out of the syringe, This will be needed to be repeated a couple of times as you push 5 milliliters of saline into that bottle. The pressure is going to build up more and more, and take it from me, you don't want to just yank the syringe out after you've just forced all that product in there. You'll end up spraying yourself. So push it till it starts to resist, and then repeat the process. Now I'm going to stir those exosomes in a circular motion like so, and we're just going to keep repeating that again and again. So I'm going to push the syringe in. I'm going to pull a little bit of that pressure off, and then I'm going to off gas the syringe. We'll push that extra air out again, and then we're going to push it back into the syringe. We're going to repeat this until we get 5 milliliters of saline completely into the bottle itself. And now we're just going to stir it up again. Make sure we do that circular motion, get them nice and stirred. So once that's done, you need to empty the syringe, so blow it into the sink, and then stick it back, an empty syringe back into the cap, flip the cap upside down, and draw all of the product out of the syringe, out of the vial, back into the syringe. You can see it going in there. So now we have exosomes mixed with saline. That is what we're going to use to put into your nebulizer, which you should have washed ahead of time with either alcohol and then rinsed with water, or with witch hazel. Either way, make sure if you do alcohol with water, you let it air dry for a bit. Put the cap back on, push the button, and you can see the gas coming out, and then you're going to place this over your mouth and nose, breathing in through your nose, out through your mouth if you're looking for brain, or in through your mouth, out through your mouth if we're trying to push more through the lungs. That's how you do it.